to see some things for the first time. And there'll be people that know a lot more about this than I do here, but hopefully some of the slides and illustrations will help for some of your lectures when you're teaching this. So the first question is, why is Paul coming to the Connectome workshop and talking about AI in neurology? And the reason is, all of you that are looking at connectomics can use this incredible set of applied mathematical concepts and ideas that have been emerging in the last 5-10 years or so. So it's a little bit like we're doing a guided tour. I'm going to take you through some areas. Maybe some of them are going to be new to you. Some of them it'll refresh and stimulate some ideas. So a summary of today's talk is we'll talk about how AI is used in neuroimaging today. What are the main methods? Maybe some of these are familiar to you. Convolutional neural networks, vision transformers, variational autoencoders, generative serial networks, diffusion models, which are very ritual, vision language models, and also federated AI. What should I be careful of? There's some things with machine learning and AI you have to be careful of. Shortcut learning, domain shift, fairness. And there's some hot topics for us in medical imaging and neuroimaging. There's the promise of automatic diagnostics and explainable AI. Generative AI is not just for making cool videos from text. You can use it for disease detection or QC. We'll show some examples on tractography data where it's used for QC. You can also use AI to enhance images or do cross-modal image synthesis, maybe PET scans from MRI. You can also use AI to merge information from multiple data types, imaging, genomics, and text. You might know about vision language models. And then we'll talk about how AI in mathematics can be adapted to other data types. Many of us are using diffusion MRI. There's something called deep normative tractometry, which you can probably guess what happens there. And also functional MRI. Can we do mind reading? Can we figure out what someone is thinking? If you had enough training data, how would you do that? So normally, I talk about this project, and I'm not going to talk about it, but there's a problem in this project. So just briefly, we run a consortium called Enigma, which is an international analysis of brain scans and genetic data from 200,000 people, 45 countries, 33 brain disorders. And the data is here, it's not centralized in any one place. And a lot of the work publishing with this has been studying what factors help or harm the brain, leading to the largest neuroimaging study of schizophrenia, bipolar, major depression, PTSD, substance use, anorexia, OCD. These are all sorts of disorders that have signatures in the brain or patterns of change in the brain that are of interest for diagnosis. And you can just say, just using mass univariate statistics, let's measure the cortical thickness of different parts of the brain and show me where patients with these diseases differ from healthy controls. And there's major depression, bipolar, schizophrenia, alcohol use, obsessive compulsive disorder, ADHD. The scale is in standardized units, so it's how many standard deviations below the typical healthy people are on average with these disorders. You can get a t-shirt if you want a t-shirt with the data on. And just to sort of say how people would do these, they would segment regions in the MRIs. FreeSurf, will give you the thickness and volume. And then basically they would meta-analyze the differences between the patients and the controls. And so you can see different diseases, schizophrenia, a lot of differences, genetic syndromes, a lot of differences, epilepsy, maybe one standard deviation difference between the quality thickness in the patients and the controls. Autism is really interesting. I know Dr. Gilmore will talk more about this tomorrow. It's one of the few disorders with more brain excess of brain matter. But if you start to look at these patterns, they're sort of signatures of the mean pattern in the different disorders. You come across this issue. So can you write an AI classifier to detect these diseases? Well, if you pick the best measure in the FreeSurf cortical thickness table, the one that showed the most difference between the patients and the controls. For something like Alzheimer's disease, you could write a classifier with that best single measure, and it would get an area under the curve of AUC of 0.92, which is pretty good, not making many mistakes. Because the difference between the Alzheimer's patients and the controls is about 2 standard deviations of the control sample distribution. In anorexia, which is the most severe of the psychiatric conditions we've studied, there's about 1 standard deviation difference. In schizophrenia, about half a standard deviation difference. And again, this is the best measure, best really right measure. Depression, 0.2 standard deviations, anxiety, 0.1 standard deviations. And so this idea of doing disease classification, where you convert the Cohen's d, the group difference, into a classifier, I mean, that's just a simple Gaussian classifier, it's not going to do very well. I mean, one measure, there isn't a good measure that's going to decisively separate these classes. So that's kind of motivating this. And so if you agree that there's no one measure that is very diagnostic on its own, I mean, what about, I mean, you don't have to use AI, you could do multivariate statistics, but if you do want to use AI, you could say, well, let's not use one measure, like a blood test as one measure. We'll use DNA, omics, imaging, which we'll talk about, maybe real-time data, integrity data, and we'll figure out which one of these data sets is the most predictive for a number of tasks. And the tasks might be diagnosis, I mean, the ones we've been talking about. Could you diagnose bipolar disorder relative to depression from a scan? It could be prognosis, I mean, future prediction, or it could be treatment response. I mean, if you have a whole lot of data on people that didn't respond to a treatment, I mean, maybe we could look at the scan and have a kind of posterior probability of that person responding. So there's kind of 2 groups of people. So there's people that want to use AI methods to actually make these decisions. So if you were a radiologist, you say, well, what's the best method? And they don't have to agree with it. I mean, they can just use it to get an opinion. So that would be someone who is using this to make a decision. The other way to use it is to discover features that help make the decision. And so if you were a geneticist, I mean, you're not really making the decision, or probably not, but you want to look over on the input side, which features ended up being helpful, and maybe we'll collect more data on them or build models around them. So there's sort of the intellectual and engineering aspects to this. So apologies to the people that teach this, but I mean, this is kind of the lay of the land. So the deep learning methods, which are essentially a way to convert input data to output predictions using a mathematical function. And the network there is essentially just a mathematical function of the inputs. It's just that you learn what the parameters of that mathematical function are going to be. And some of the classical ways have come from computer vision. So let's say you have a new lab member, she's called Sarah, you want to make sure she can get into the lab. And there's lots of images of her and other lab members, and you train a convolutional neural network to identify it. And this comes from biometrics. And basically, multi-layer feature extractors will identify consistent features about that person's face. And by analogy, if we change our faces for MRIs, I mean, we're not saying this person gets into the lab, but we might be predicting whether they have a diagnosis of Alzheimer's or some other pathology. Or you might be benchmarking. A lot of people might predict the age of the person, something called brain age. And we're not interested in their age, we could just ask them. But it's something with ground truth where you could train the algorithm to see how well it does. And you could say, does this person have Alzheimer's disease, where one treatment is applied, vascular disease, where a different treatment is applied, or something else? Are they amyloid positive, and do they have enough amyloid in the brain where they might be eligible for a trial of a drug that clears it, like there's a new drug with canamide for Alzheimer's disease? So these have a lot of applications. So my first grad student, there he is, Alain Petiot, was working in 1998 with neural nets for finding the corpus callosum in MRI. Nowadays that's easy, it's solved. I think it's solved. So his other collaborators at INRIA in France didn't like this. They said, don't use neural networks, it's really bad. They disliked artificial neural networks. They said, well, how can we see how it works? And they said it has millions of parameters. And in modeling, you want models that have few parameters. You won't be able to understand the models. And others of us said it will never work. And actually, it didn't really work. I mean, it sort of got the corpus callosum, but bits of other things that are not the corpus callosum. But he was actually very discouraged. I mean, most of the community did not like deep learning. Just to clarify, this isn't really deep learning how we do it today. So the features, like Gabor filters and Hilbert experiments, they're mathematical features that he picked. So there is a type of deep learning that uses radiomic features, where you pick the filters going in. Normally today we'd learn these filters. We estimate the powers of filters going in. Now, flash forward to today, you can train a convolutional neural net to detect Alzheimer's disease. So Xiaogang Yan and Bin Lu at the Chinese Academy of Sciences essentially fine-tuned inception ResNet, which is a state-of-the-art CNN, on 85,000 scans from 50,000 people. About a third of them with Alzheimer's disease, about 2/3 that were controls. And they basically were able to classify Alzheimer's disease with 91% accuracy, which is about as good as it gets because the diagnosis has a little bit of inter-reader variation. So if you see 100% accuracy, a little bit suspicious. So how does that work? I mean, it's kind of an interesting thing. The other way it can work is AI can be used for subtyping dementia pathology. So I can see this person has dementia, my memory has declined, but I don't know whether it's due to amyloid or vascular disease, and the treatments for those are different, or maybe it's some other thing. So Guga Toastman has trained classifiers on neuropathology data sets from people that donated their brain to science but were scanned before they died. And if we only have multimodal imaging when they're alive, we'd like to say, do they have amyloid or Lewy bodies? Lewy bodies are present in Parkinson's disease, or TDP43, which is a tau pathology. And this is about, it's an N-way classifier. This is about 86 to 89% accuracy. And the reason you do this is that amyloid trials only want the people here. They don't want to treat people that have a different pathology. But you can't tell. I mean, it might be a new type of PET scan that would help, but the main scans cannot tell. The other one is, this is some recent coverage, is AI that reads brain scans shows promise for finding Alzheimer's genes. How does that work? Well, if you run an AI disease classifier on 100,000 images, test images, you could then run a genome-wide association analysis on its output to discover genes that affect the disease, and people are doing this. So let's say you feed in PET scans and a CNN finds regions in the image that help you predict Alzheimer's disease. You can then go ahead and feed the output of a classifier into a classifier that's been trained on genomic data, and go and find genomic markers that promote the disease. And I mean, you can let it loose on all the data you have. I mean, you train it on some data and then go and discover. So this work by Taeho Zhou in Indiana, and his team in Indiana and Korea, are basically using AI to screen massive databases of images and genomes and go look at the genome, which is kind of cool. So when the students are trying one of these problems, let's say that the student says, I want to write a classifier that detects autism. The first question is, will we ever have enough training data? Do we have enough training data? And to make the problem more well posed, to achieve a certain performance level X, we have enough data. And so you could study how does deep learning performance depend on the amount or diversity of the training data, maybe the GPU type, maybe you need a lot of training time. And this depends on the task. It depends on the model architecture. CNN's vision transformers differ in their inductive biases. Depends on the ingenuity of the developer. I mean, obviously in mathematics, as different people attempt a problem, the mileage may vary. So we're going to talk about pre-training neural networks, foundation models, generative pre-training. Also, people are looking at how the training is used. So curriculum learning or adaptive learning or reinforcement learning, they don't just pass the training data through the classifier. They do different things. They pass it in a certain order, or they pick certain examples that are either very difficult or very easy. And you also want to correctly avoid overfitting and confounding. So you could have a fabulous result, but if it's locked onto a feature that isn't generalizable, it won't work in practice. So let's compare AI methods. So this is accuracy in detecting Alzheimer's disease. And as there's more training data, the accuracy goes up to about 91%. And I'll fill in the names of these methods. So this is a paper Nikhil Dinakar and our group. He was evaluating deep learning methods to take standard MRI scans from Alzheimer's patients and controls and feed in different numbers of these as training data, and then an independent test data, how well did it do in classifying Alzheimer's disease. And there are old-fashioned radiomic methods, 2D CNNs, and fancy 3D CNNs. And I'm going to define some of these different things. But the best methods, the one that did best, we'll talk about what it is about those methods that make them better than the others. But obviously, to be useful, it should be better than about 90%. So a lot of computer vision and AI language models are benchmarked, like the standard data sets. We don't really have that in medical imaging. So this is work on video and synthetic MRI pre-training of 3D vision architectures. And this again is an idea where you could benchmark dozens and dozens of AI networks to detect Alzheimer's disease and related dementias. In this study, the best performing models were 27 million parameter swim transformers, they're sliding window vision transformers. And you can say, well, is that true all the time? Is a certain mathematical architecture generally better? Or is it sort of, no free lunch? I mean, maybe different architectures for different problems. And so in language models, you'll have heard of something called superglue. It's a benchmarking system. And if you've developed a language model or fine-tuned one, you have to do a whole lot of tests, like sentiment analysis or sentence completion. And so this would be kind of cool for medical imaging if we're sort of benchmarking data set. And people are earning a lot of money with this. So the FDA has approved a bunch of these methods. BrainSeed, which I have nothing to do with, raised 10 million in venture capital, and then the Alzheimer's detector. It's actually, I mean, those of you that publish at MICI and ICML, it's only as good as the papers. I mean, they just went and raised money and productized their development. So these things don't have to be absolutely perfect to be used, and a lot of the ones that are used are not perfect. And the radiologists find them. They want a second pair of eyes. So let's take a couple of steps back. So again, apologies if you teach this, but it's also kind of fun and new. So in a standard convolutional neural network, you're going to feed in the MRI, and the MRI is 3D. And each successive layer of the neural network applies larger scale filter kernels, or you can think of them as feature detectors, to distill useful features from the previous layer. And you can basically write this out as a mathematical function. And so each neuron in the neural network computes a function from its inputs. The parameters are initially set to random values, and we adjust those parameters during training. And there's some cool videos, I'm sure you've seen these before. Usually the weights are updated using stochastic gradient descent. You take a batch of training data, you figure out what the loss function is at the end, and you tune the parameters of the neural network such that the loss is reduced. And if you compute the error and adjust the weights by a backpropagation, this is repeated many, many epochs. An epoch is one transit of the training data through the neural network, until the validation set, which is a separate set of data you set aside, until the validation set loss, how badly it's doing, no longer decreases. And then you freeze it. And you say, I've trained my neural net, let's other people test it on their own data. And this is kind of fun. I borrowed this video. So this began as a digit recognition problem. So when I was a graduate student, there were no computer vision methods that could recognize handwritten digits. And mail went to the wrong place. I guess if you didn't write the zip code well. So Jan LeCun, and I mean many researchers solved this by essentially, this is a CNN, essentially embedding the digit geometry into an embedding space, and then developing mathematical functions that would do an n-way classifier, in this case a 10-way classifier. And these are just grayscale images, or they could even be binary images. It's not a really challenging problem, but this showed that CNNs really could work. And we are interested in kind of doing a little bit like digit recognition. I mean, let's say here's some epilepsy patients, some healthy people, some Alzheimer's patients. Let's say you have 10 diseases. You can say, well, can I just use Gann-LeCun's digit recognizer and put in the MRI scans, and it will just all the time tell me what disease the person has. I mean, you could try it, it's kind of interesting. And you know, the features detected in these scans, the top one is the first convolutional layer, second convolutional layer, third convolutional layer. They will learn to see features at different scales and different layers of the neural network as they learn to do the classification. And you can look at them. And so, I mean, this is a cool book. If you have students interested in explainable AI, that book's kind of good. It talks about occlusion sensitivity analysis, GradCam, layer-wise relevance propagation, and if you're doing vision transformers, there's something called attention flow. These are all ways of saying, well, what did the AI method use, or what did it rely on in making its decision? So if you have epilepsy, you can have seizures in your left temporal lobe, right temporal lobe. You can't really tell on an MRI all the time where the seizure focus is. But if you train a classifier to say which side is the seizure focus, and then use these techniques to say, well, tell me exactly which areas you found helpful, it is kind of cool. And this is ongoing work by Eric Kastner on epilepsy. So if you haven't seen these before, it's very simple. Once the CNN is trained, you cover up some of the image. This is an approach called occlusion sensitivity analysis. And if you systematically loop through the input data and you cover up some of it, if a certain area is really important to making the classification, then the error goes up. And you could essentially make a map of the sensitivity of the output to covering up various parts of the input. And these are sometimes called saliency maps. So let's look at the brain on the right. If that temporal lobe region was really important to deciding whether you have Alzheimer's disease or whether you have left temporal lobe epilepsy, but if you have left temporal lobe epilepsy, but you covered up the left temporal lobe, presumably that's not a good thing. So NY classifiers can create an occlusion sensitivity analysis map per class. And what it's defined as is the gradient of that class's output probability with respect to the occlusion of each input layer. Now this doesn't always work. So some neural nets are trained using dropout. So you actually train them to make sure that they're not sensitive to the occlusion of any of the features. So that it will be blank if you train it with dropout. But this is kind of cool. It works. There's some fun examples. If you train a neural network on videos, and you say, I'd like to watch a video of boxing. Maybe you're watching the Olympics. The saliency map in the boxing video shows the person boxing. We know where the person is. It's kind of cool. It's found the thing that makes it most likely that that's a video about boxing. And you could speculate, is it the red gloves, is it the person? But I mean, it's done a good job. We didn't tell it that that's what happens in boxing. It just learned by itself. The other way to do interpretable AI is to cover up the later layers of skin. So rather than the input, you can cover up the output or later layers and see which ones matter the most. And this is cool too. So if you train a classifier to recognize cats and dogs, and you say, what is the gradient of the output class probability with respect to including the later layers when it sees the cats, the dogs, and you guys have seen this. It's actually kind of good at difficult things like brushing teeth. I mean, this woman was brushing her teeth and somehow figured out that those are the features. And this gentleman's cutting trees, and I don't know, the chainsaw gets highlighted. It's kind of cool. If you apply these to MRI, they're good sometimes, but not always. So if you train an MRI classifier to find gliomas and tumors, I mean, it found that glioma, that was pretty good. But I don't think you'd be very pleased if you had a pituitary tumor and it said, well, hey, there's something in the brain. I mean, the reason is it works, but it's limited by the spatial resolution of the last convolutional layer. So multi-layer GradCam, and this is used in resting state connectomics. You can say, let's train a classifier to identify a disease based on resting state data. I mean, these are network inputs. And in this work by Vince Calhoun, he's looked at the saliency map for every layer of the convolutional network. He said, I'm going to input resting state fMRI and classify schizophrenia versus controls. And I wanted the grad camp to tell me where in the resting state data it was helpful in deciding whether someone had schizophrenia or whether they were healthy control. And the first convolutional layer found the edges of the default mode network. It found the edges of it. And then the second convolutional layer found the middle of the default mode network. And Vince Calhoun, who's an expert in resting state, said, well, that's kind of cool. That is one of the signatures that we use to decide whether a person has schizophrenia based on their imaging. So let's talk about the CNNs themselves. So we've talked a little bit about how if you keep having training data, the accuracy goes up from 0.65 to about 0.9. Let's say you pick the feature. So let's say you use local histograms or Gabor filters or your favorite feature detector, but you freeze those and decide them. You'll get about 72% accuracy in classifying Alzheimer's. And these methods are not really as good. You can use 2D CNN. So let's say an MRI is a 3D, but let's say you just give it a 2D slice. Maybe there's enough signs of the disease in 2D slices. So people have written ResNets which handle 2D images and concatenated them using a long short-term memory architecture, which is used in sequence AI. So this is, LSTMs are often used to process text. And you say, when we have a sequence of decisions from the 2D images, could you tell me overall what the chance of the disease is? And these work okay. They do better, 78% accuracy. They do better than choosing the features. They learn the features, but you're only looking in 2D. Maybe out of plane there's something interesting that this isn't seen. Or you can use 3D kernels. So one of the best methods that 91% accurate on this Alzheimer's test. You basically take every layer and concatenate them at each level. So you have a sort of multi-scale, a sort of octree grid. This has been used in computer vision forever. That does really well. The connectivity of the networks is a little bit different. The straight CNN has successive convolutions. Output is better than next layer's input. ResNet has some skip connections that were introduced to avoid gradient blow-up or gradient decay. And DenseNet has loads of skip connections. It actually feeds data from the beginning to the end and to all of the intermediate steps, and that seems to help. Now, a rival to CNNs are vision transformers. And so they, transformers usually input language tokens, words, and they either classify the text as happy or sad, that's sentiment analysis, or they continue the text. But you can feed in those tokens patches of images. So vision transformers take little, usually 16 by 16 pieces of images. And amazingly, this does work for MRI classification. It outperforms CNNs when the amount of data is large. So vision transformers are a little bit more data hungry than CNNs. But it outperforms the radiomics method. And we'll talk a little bit about this efficiently training vision transformers on structural MRI. For Alzheimer's detection. Now, just a reminder, most people don't train these methods from scratch, so they go get a CNN or a vision transformer that someone else has trained, usually on photographs, natural images, and they do something called fine-tuning. So these diagnostic CNNs are often pre-trained on a larger data set like ImageNet, which is 14 million photographs of things. And then the weights are frozen in the early layers, because presumably you have low-level features that are always useful. And then the weights in the later layers are fine-tuned on the medical test. So this instrument is really good. Actually, this one distinguishes COVID from viral pneumonia. They took an ImageNet trained classifier from the internet, and they only tuned the last layers. And so that's kind of a standard thing to do. And you can say, wait a minute, I don't know what ImageNet is. So for those of you who don't know what ImageNet is, Feifei Li at Stanford in 2012 had people annotate photographs. So there's the Norwegian flag, there's various cups and eggs, including some things that I don't think we could tell what it is. And AlexNet, which was an early CNN, very, very deep CNN developed by Alex Krzyzewski, really revolutionized this field. And these are the features it sees when it's been looking at photographs. these aren't really objects in the first layers, they're low-level features that it was learned. But there was a competition, and every year you could enter the contest. And if your CNN was able to correctly identify the objects in the photographs, you'd be on a leaderboard. And so it'd be like a scoreboard in 2014, AlexNet, which looks like this, was winning. And then people had this model, they could change it. I mean, they could adapt it and add new units. And then there's Inception, the one that we talked about a little bit earlier. And this keeps getting better. It's not 100% accurate. I mean, it's like the MRIs. So some of these might have been labeled wrong. I mean, maybe the asymptote is lower. So you could say, well, all right, in terms of accuracy in identifying things, and the more compute time to train it, and then the bigness of the dot is the number of parameters in the neural network. These are things that your students could try. I mean, most of these are available. And you say, well, I'm going to take one of these, I'm going to freeze the early layers, I'm going to show it some MRIs or some connectomes, and I'm basically going to see how well that adapts to medical data. And a favorite one is the VGG means Visual Geometry Group, and this is one with 19 layers. A favorite one is VGG 19. This one's actually used in a lot of medical image analysis papers. And if you have an input that's 224 by 224 by 64 coming in, I mean, it will make an n-way classification if you train it. So the decisions to make if you want to train CNN to detect disease in a 3D brain MRI are these. One is brain MRIs are 3D, or if you do fMRI, they're 4D. Should we stick with 2D CNNs, or should we use 3D CNNs with 3D kernels? So that's going to need more RAM. It might mean smaller mini batches for training. Transfer learning, should we just use ImageNet, which has over 1 million, actually 14 million 2D photographs? Or should we train it from scratch on other large brain MRI data sets? So UK Biobank has 46,000 MRIs. It's not 14 million. But what task should we train it on? You don't have to pre-train it on the same task that you're going to do later. And some people do inferring the age or the sex of the person as a pre-training task. And then there's another approach, contrastive learning, which is a self or semi-supervised learning technique. You first embed the images into a much lower dimensional latent space, where similar examples are close together, and use this feature embedding into incidental classification. So just to sort of talk about that, a lot of the best CNNs have been pre-trained using self or semi-supervised learning. This is the original paper on Sinclair. But what you do kind of makes sense. So if you have a control image, maybe if that was brighter or darker or rotated, I mean, there's a whole lot of augmentation transformations that make no difference to the classification probability of that image. So you will augment your training data set in all sorts of different ways. There's sort of an equivalence class that you want to mod out when you do the classification. And then in the embedding, you design a triplet loss or an n-pair loss that puts all of the augmented images of the same thing close together, and all of the other examples further away. It's sort of like tidying the room. If you put all the socks over here, and you put all of the shirts over here, it's doing some pre-tidying of the embedding space so that the equivalence class or orbits under the same set of invariance transformations are not interfering with the output. So that's cool. Who here has seen this? I'll play it a couple of times so you can see this. It's kind of fun. You see a brain emerging. We'll do it again. So does anyone here know what is going on here? If you've been to Mackay, you probably know what this is. Yes, what is going on?
Is it the diffusion process?
It is a diffusion process. So we took noise and made a brain. It looks kind of strange. And the brain looks good. Very, I don't know, it looked like a brain to me. You can make a brain with known ventricular size. That one in the middle is silly. No one has ventricles over here, not many people. But you say, I'd like a brain with ventricles that occupy half of the image, or a brain with no ventricles. I don't know if there's anyone with no ventricles. So a latent diffusion model can generate brains conditioned on age, sex, ventricular volume, brains. I mean, you can make brains. I mean, you can train things on brains of a certain age. I mean, assuming it's getting it right, we'll talk about how it does it. And this sort of nightmare diagram is how these work. But this is a model by Walter Panaya. It's a really cool paper at Mackay in 2022. where they trained a so-called latent diffusion model on basically all of UK biobank, or nearly all of it, 31,000 T1 weighted MRIs. And how does it make brains? And I want to say why you would do this, but we'll say how it does it first. So if you ever go to this workshop, it is super cool. There's like 4 hours of a tutorial on diffusion models in medical imaging. They're super cool. But I'll do the 5-minute summary of the workshop. So a generative AI model is different than a discriminative AI model. So the discriminative AI models we've talked about so far took inputs and they condensed that down to a decision, like the class probability of Alzheimer's disease. A generative AI model will make more data that resembles the training data. It'll make additional images in our case that have a certain property that we've decided how it should look. Now, an autoencoder, the property that we're deciding how it should look is that it looks the same as the input. So why do I want an image that looks the same as the input? It's a special neural network with a bottleneck. And so a very short autoencoder has a lot of inputs, all the pixels if you like, but it only allows what's called a bottleneck layer, a layer with a very small number of dimensions to be put in the middle. And then this part of the neural network, which is the decoder, tries to reconstruct the original image. And you keep passing training data through. If it does a bad job, the parameters here are tweaked so that it does a better job. So it's a data compression technique. It's kind of like JPEG. It's a data compression technique. So if you do this and you use a 2-dimensional latent space, so if you have a big high-dimensional image set and a 2-dimensional latent space, you'll find that it places all these crazy inputs into an organized matrix, so that when they're embedded in 2D and you just dial up one image of that there, certain digits with the same morphology are next to each other. And you can imagine how you could generate digits. You could go to a latent space, use the trained decoder, and go and make a digit of that kind. So essentially, a standard autoencoder will put all these digits in discrete areas of the latent space. Here the latent space is 2D, but it can be high-dimensional space. And all the number 7s go over here, the number 8s go over here, the number 9s. And many of you have used UMAP or TSNE or even PCA. I mean, there are ways to get high-dimensional data into a lower-dimensional space. And we're going to be using those to separate classes. We're going to be doing things that help us organize the data in a way where the classes are separated. A variation autoencoder is a slight tweak on the standard autoencoder where rather than just put these data in patches, the output of this latent space layer is supposed to be as close as possible to a multivariate Gaussian, multidimensional Gaussian. And so it just has an additional constraint that this embedding has to put this into a sort of Gaussian cloud. And that's easy to sample from. If we generate a random Gaussian vector anywhere here and go make an image, you can make new digits. And this is kind of fun. VAE training forces the latent variables to compress your input into an embedding space. You learn vectors U and sigma, which are nonlinear functions of the input data points. And then when you input a training data point, it lands in latent space in a certain position that's in this multi-dimensional cloud. It's like you've made your own cloud of latent data. And the training loss per data point for the decoder and the encoder model network parameters is the sum of 2 terms. One is how closely do you embed your latent data, how close is it to a Gaussian? So the symmetrized KL divergence is measuring how well you're doing at each training iteration. And then L2 loss, mean square error loss, is telling you how well you're reconstructing data. Now, all you're doing is compressing data. You're trying to compress it in a way where the compression has a nice multivariate normal density. And if you feed in images that have been covered up, I know that's somewhere in Venice and someone's covered up the middle of it. How do they do that? So the output got filled in and it makes sense. Well, look at this. So here's a photo. Let's say that part was covered up. How did it do that? I mean, it's kind of an amazing thing that it sensibly fills in the speed mode. And those of you that have tried PCA, don't do that. I mean, the VAEs are kind of special that they'll fill these things in. And if this makes you think of large language models and mass token prediction, you're exactly thinking in the right way. So, why does this look so much better than PCA-based inpainting or imputation? And if we have missing data, and someone said, could you please impute the missing data? I mean, how does it know that this band is playing? I mean, they're missing. How does it know that this particular band? It's guessing. So one of the breakthroughs in this field was to add an extra reconstruction loss term when training a VAE. And so, essentially, if you were training on faces and you were making a generative model of faces, and you wanted to occlude a face and put the correct thing back in the place where the face was, this cool paper, Deep Feature Consistent Variation Autoencoders by Shanshu Hu, adds a perceptual loss to the reconstruction from an L2 norm on all the features detected by L layers of the VGG19 CNN. Now let's unpack that a little bit. Remember how the VGG CNN can identify objects. So if you pass a face through it, it goes, oh, it's a face. If you pass noise through it, it doesn't know that that's a face. So we can ask VGG CNN to look at the input and tell us what all the features are, and make sure the output has VGG react in the same way. A little like a gap, a little like a gap. So if you put the VGG on the side, have the inputs go through it, take the feature extraction layers of the VGG CNN, and then at the output of the variation autoencoder, you compare VGG's opinion of the image. It'll make a really good image. And so it's kind of a revolutionary idea. I mean, if you do VAEs trained with pixel-wise loss, they're really blurry. I mean, it's a compression, especially 2D embedding. This is going to be awful. But the VAEs trained with this added perceptual loss, look, you can't really do hair. I don't know, this person has see-through hair. But it does faces really well. So the idea, you can see the difference. So standard VAE marker, VAEs trained with perceptual loss, they look like people cannot. So basically, if you embed faces with a perceptual loss VAE and look at the latent space, we're looking at a 2D latent space. You can use 20 of these now. You'll see faces looking right over here, now I'm doing that. Faces looking left or over there. There's some blonde people. This paper explains it. It's kind of a cool thing. So you can see how we're going to be generating brains in a minute. It works with faces, which are already pretty hard. Now, a VAE can generate, using the decoder, the trained decoder, images corresponding to a random vector. So we're going to put a random vector in the multivariate Gaussian space into the decoder and say, can you make me a face, please, that has this vector as its latent code? And this is kind of cool. It's called Visualizing the Latent Space. This video will change how you imagine neural nets. You can play with this. You can make faces. It's kind of fun. So if you do this with brains, you'll get brains that vary gradually along hundreds of natural dimensions of variation, plus synthetic brains in between. So let's say you embed data in a latent space, and you compute the brain volume, and you make a cloud in the latent space where all the brains are with a certain brain volume. You can now sample from that latent space and use the trained decoder to make a brain with a normalized brain volume of a prescribed value. And Walter Panaya's paper takes you through the steps. And he provides code. So what's nice about this is your students can download the code, play with it, train it on new data. This is trained on UK data, which is really good data. But actually, the radiologists say the brains look great. I mean, I've been looking at brains for a long time. The problem with earlier models is it didn't get the cortical thickness right. And those of you that are specialists in GANs or deepfakes or things like this know that there's a function called the minimum mean discrepancy. It comes from reproducing kernel Hilbert space theory, where you say a way to tell whether this is fake or real is to develop a set of test functions. So you have those of you familiar with functional analysis, you have sort of the integral of a test function, a feature detector, convolved with the image, and the infimum over the group difference between fake and real in that function is estimated. and you estimate it over a class of all allowable functionals. And this is a way of formulating deepfake detection using functional analysis. And I think actually the results look pretty good. And you could say, well, I just don't want a brain, not useful to have a new brain. Can we condition the models and generate brain images where we can specify the age, sex, ventricular volume, and brain volume? Now, there isn't one brain that has a certain age and sex, but there's a distribution of brains that are conditional at age and sex. And so there was a paper by Rombach that shows you how to do conditional latent diffusion, which was used in this paper on brain generation. And you can do some fun things. So let's say you train your latent diffusion model on healthy brains, and you put in a diseased brain, and it hasn't seen any diseased brains. The best it can do is reconstruct the closest healthy brain in the latent space. And you can actually crop the multi-dimensional counting a little bit as well. And if you subtract the autoencoder's best healthy brain from the real brain, you can argue whether that's the anomaly map. It's probably the anomaly map plus error, plus a lot of other terms. So it's kind of a normative deep learning model of anomalies. Now these models have been evolving really, really fast. So this video, so this software is Quaishu's clean software. And someone said, please show a video of a gentleman eating noodles. And the VLM makes a video, and here's a little boy eating a hamburger. It does a really good job. So these models for video generation use the same concept of VAE to amount the image data or video data, but they use a diffusion process on probability distributions. They use the Fokker-Planck equation, which is a way to evolve a probability distribution of time. And they're basically saying there's real data, and we'd like to take the embeddings of data and stretch that data using a flow or an evolution equation so that we can model the real data, very complicated data, and then sample from it. And you can use text or a vector classifier guidance for conditional data generation. So in this context, it's a prompt. A text prompt says what to generate. In our context, it's a vector. You could say, I'd like a brain with a certain age, sex, and other characteristics. And just very briefly, I'll tell you how this works. If you haven't seen these before, they're super cool. So denoising diffusion probabilistic models, which are a type of generative model, take noise. These are photographs. They add Gaussian noise, a variance that depends on t, and they turn it into Gaussian noise. And then DDPMs learn to invert the noising process, and so they, So this is early work by Yasha Solhekstein, which was, I say early work, it was 2015, which ran an ODE in the opposite direction and found out the gradient of the current image, the direction of change of the current image, that would make the log probability of this being real the highest. And so essentially this PDE, which denoises images, learns from it's called a score function. This is something that's called the score function. It's the gradient of the probability density of the current image in sample space. And this works. It's cool. You can take a training set, add noise, train a neural network to denoise the image and make it look like real examples. And I encourage you, anyone that's interested in mathematical statistics, it's a super cool area of theory, which I cannot completely get into. DDPMs in data space are adding noise and removing noise. But if you think of the latent space where all this data lives, there's a latent space, an embedding space where all real data lives. And you're essentially trying to morph a multidimensional Gaussian using a flow to stretch it onto how real data looks. And then in this real data sort of morph space, you're trying to find certain areas that have a certain age or a certain sex. You're traveling through this latent space and sampling, using a standard sampler to get the images. So essentially, if you want to create images that look like an Alzheimer's patient, you do latent space denoising using this score function method. you have an auxiliary classifier. What is the likelihood of being Alzheimer's class at time t, and you add some Lagrange multiplier of that. Now, I want to make a distinction. Unlike classifier training, where we obtain gradients of the weight parameters for updating by a gradient descent, here we compute the gradient with respect to the input image. So we're changing the image. The image is not very good, it's noisy, and we're calculating the change in the image to make it have a certain property, like an Alzheimer's patient or a 10-year-old or something like that. And the theory of this is just fantastic. I mean, all of you who are applied mathematicians, you would really enjoy this. As well as noising and denoising training data, there's Fokker-Planck equation is a way of thinking about how this model data distribution is evolving through time using essentially something called Langevin diffusion, which many of you will be familiar with. So these are cool. I mean, one application is you make the closest healthy image to your patient and subtract it. And VAEs, we said, could do this. But everything looks abnormal. Why does everything look abnormal? Well, it didn't approximate that person very well. But these DDI, they're actually pretty good. I mean, this is someone with a tumor, and they highlight. And you can have radiologists do nice coefficient stuff to correlate this. So last 5 minutes, we're going to have a recap of the last 5 minutes. So generative AI can simulate the reversal or progression of a disease on the individual's own brain. Now, many of you will be familiar, I know Tom, you're very familiar with this, with LDDML. So for the last 20 years, there's been something called large distance diffeomorphic metric mapping. And it's a mathematical formulation that describes the warping of images. And so if you were measuring growth or atrophy, or if you just want to register data onto a template, there's some smooth or differentiable mapping. And you'd like this mapping to cause your image to match a template both in intensity or some mutual information property if it's a different modality. Subject to the regularization condition that it's the least energy mapping, that the flow doesn't cost a lot, like optimal transport. And it's an optimal transport. Or a continuum mechanical operator that's describing the energy of the mapping. So you could say, how does latent diffusion modeling relate to this LDDMM framework that has been used for registration for a really long time? And there's a lot of interest in this. So I'm presenting this as if it's all solved. It's not all solved. There's a lot of open questions in this field. One of them is, how are these generative models validated? So you can use within subject longitudinal data to verify the progression or the interpolation. LDDMM also does this type of thing. It will interpolate or extrapolate images using geodesic shooting. You can test if the images are fake or real. I mean, how realistic are they? So tests of realism, tests of the generative distribution resembling the real distribution, usually you use GANs, you use an adversary to claim features that discriminate them. They use families of divergences. A nice one is MMD, which has a nice closed form formula for discrete points. And I encourage you to look at work by Arthur Gretton. So he's an expert in reproducing kernel Hilbert space theory. And he has a whole lot of cool ideas about GANs and generative model, really, really amazing, that will help kind of simplify in your mind why different choices are made in this field. Now you can use this for connectomics. So I mean, I haven't shown you any connectomics examples, but you can generate resting state data. Vince Calvin said, well, you need to show some connectomics. So his ICA pipeline will allow you to create under certain conditions, resting state data that obeys certain statistical conditions. You can also apply these generative AI methods to diffusion connectomics. So let's say this data comes from, it's actually from Camille Ogerville, who's going to speak this afternoon. This is scan 70 pointers. So all those streamlines can be fed through an autoencoder. And did you notice how they're kind of messy? But then after they've come through the autoencoder, they look nice. I don't know, maybe it already does some good stuff. We're going to make sure that that's doing it right. But you can think of this as a low-pass filter in latent space. And you could sort of say, look, in textbooks, these connections look fabulous. I mean, they never look better than they are in these pictures. But in my lab, they look like this. I mean, when the data comes in, I mean, is that an arcuate fasciculus? I don't know. What is this? So some of the ways to clean up data can use the concepts that we've been describing. So let's say, this is called fiber-leak. It's one of our postdocs, Brad Chandio. Let's say you take streamlines in 3D space, put them through an autoencoder. This isn't popular anymore, TC. They're okay. They're all dimension reduction methods that preserve metrics in different ways. But you can take kind of messy looking tractography data and output clean data, called fiber. And you can use these for unsupervised white matter tract filtering. You could also use the generative model as an abnormality detector. So this is Wendy Fan. She said, well, I know we've been tidying up streamlines using autoencoders, but the latent space is a generative model. I mean, you can score streamlines. You could actually say an outlier, or maybe there's some patterns associated with disease. So this is called deep normative tractography. We were not the first to use it. There's a person called Maxime Chamberlain, who many of you might know, who's been doing this. We don't have time to talk about this. We'll talk about it another day. It's kind of cool. It's a different way of doing cross-modal synthesis. We'll do another talk, but maybe next week we'll talk about it. But I just want to remind you, generative AI doesn't have to generate the same modality of data. You can make PET scans from MRIs. I mean, you can learn cross-modal mappings. I mean, that video of the man eating a hamburger, that's very cross-modal. It's text to the video. So, I mean, this should be, now, I don't want to get too cocky about it. There has to be some mutual information between the target and input modality. But if these data modalities are irrelevant to each other, you're not going to be able to learn embedding or any cross-attention between them. Last one. So Xiaoyan Yan is using AI. He works on depression, on fMRI time series. And just like ChatGPT or other language transformers, he does mass token embedding and uses the transformer to fill in the mass token. And using a sort of autoregressive approach to fMRI time series, he can predict future time series. Not very well. I mean, you can't predict fMRI beyond a few milliseconds. But once the encoder has been trained, he uses that to classify mental states and the percent of time a person is in each mental state. So you can be either daydreaming or depressed or regretting something or happy. And you can imagine this readout could be helpful in a trial of a depression treatment. This kind of thing. Okay, we're not going to talk about things to be happy. But these are cool. All sorts of cool ways to do deconfining, but that will be another day. So anyway, thanks for coming on the guided tour of AI in neurology. We talked about two branches of AI in neurology. So discriminative AI, for want of a better term, might be doing disease detection with neuroimaging, and there's a zoo of methods. You don't have to make them from scratch. They're all on the internet. You can download them and try them. There's a zoo of methods being benchmarked. So we talked about convolutional neural networks, vision transformers, and sequence AI methods like long short-term memory, recurrent neural networks. And in our context, they read a series of 2D slices of sequence and make a decision, and hybrids. Explainable AI shows the features used, so OSA, grad count, the simplest. Attention flow is kind of cool if you look at that, with major applications in radiology. Generative AI makes new image contrasts. I call it contrast because it could either be a synthetic same modality data set, or a synthetic other modality data set, which is the video of the person eating a hamburger from the text prompt. And this could be used to enhance images or do counterfactual image generation or conditional image generation. You can imagine seeing pathologies we can't see yet with neuroimaging if you have paired data. And we didn't talk about neural style transfer or GANs. We'll talk about that another time. But this general outline of work very promising. The final warning is that pair is needed to model the site or span or data set confounds. So to avoid shortcut learning, there's actually a set of methods for adversarial deconfounding. And so if you're worried that it's learning something about the scanner or the scan protocol, you can actually put an adversary in your neural network that comes from algorithmic fairness. It competes out signals from the latent space that would help to classify that scanner. And so these adversarial methods for deconfounding are very popular right now. And if you want, you can join in it. So if you want to test methods on diverse multi-site data, my day job is something else. So if you want to try and get some disease data to try these things, there's plenty. So just a word of facts. The people that collected the data for these studies are here, all the people. And the people that have been funding this is an NIH grant, AI for AD. Liang Chan, who's here, is working on with us. Again, thanks to Zheng Wu and to Goran, Yu, Mijan, for a fabulous meeting. Looking forward to learning a whole lot from you guys. Thanks so much.
Any questions? Hi, thanks for the great sharing. So I have two quick questions about the generative AI. So I'm from AI community, so forgive me if I ask some stupid questions. So basically, I think so basically, in my mind, generative AI emphasize two things like fidelity and diversity.
Yes. So
especially for the brain image generation, how could we measure the diversity? So it seems like it seems for me they look the same, the brains, but mathematically or numerically, how can we measure the diversity?
Yeah, so there's something, you know this called mode collapse, where there's a very diverse set of images. Let's say images of each of us. And let's say the generator only generates images of one person. And that would be mode collapse. It would be a sort of place in the generative distribution that the decoder focuses on. And so, the known techniques, as you know, to mitigate this is, you would use samplers that navigate the whole latent space. So in Langevin diffusion, there's a noise term that tries to push you out into these other areas. So they usually test it with adversaries. And so if there's a certain class of images that are not coming out of the generative model, an adversary can find those and then say, do a better job of leaning towards this data. But I kind of agree with your question. With brains, we wouldn't notice if a certain kind of brain wasn't being generated. That's you don't notice something that you don't notice. It's sort of a tautology. So it's a very philosophical question, isn't it? If the only information you generate is sort of this consistent set, you don't really know if there are data points. And that's been a criticism of generative brain models. Maybe they don't reach certain kinds of brain that haven't been seen. And so how would they assign a score to that brain? And I don't know if do you have any ideas for how all of us are working in related areas. Do you have any ideas for how it could be improved?
Yeah, I think currently it's straightforward way will be, as you mentioned, adversary classifying with a trend on data with different categories to detect whether certain group of images are generative or not generative. I don't have better ideas beyond that. Thanks for the question. Thanks for those.
Appreciate it.
Thanks.
Any other questions?
I think I'm ask a question about the resources. As a young researcher in the university who just started the group, those models, I also taught the machine learning class. Those models become bigger and bigger, right? How can we get resources? Do you have any comments on how can those researchers get resources to try to run a model and then do some research on it?
So Zhenglu's question, I like that question because if you were teaching a class, and you are, where the students have to train a model, either it costs money or there's not enough GPU or TPU. So I don't know. You guys are mathematicians, and I've seen a little bit of parameter efficient fine tuning or low rank adapters. There's sort of a move at the same time towards more efficient networks. So it seems like when a problem is solved, it's like math, isn't it? I mean, when you have a least upper bound on a solution in math, there can be this crazy least upper bound, and then through the process of time, people get more and more efficient networks. So some of the models that I showed today, the students are allowed making sort of low rank or PEFT, which is parameter efficient fine tuning, where you sort of have a teacher with too many parameters and a student with not many parameters. I don't know what that analogy is used. But you kind of teach a student neural network to emulate the bigger one using less resources. And I mean, you know from approximation theory that there will be some good results in that area about how close you get. So I think to give you an honest answer, people will always use the most resources they can get. I mean, it's human nature. But it doesn't mean that the problems can only be solved by people with the most resources. Because, I mean, look at math. I mean, math problems are solved, I mean, many problems are solved without, it being the person that owns the most capital to do. So hopefully that's optimistic.
Sure. I mean, it's a frustrating issue because, I heard some comments from the industry saying that for people in academia, you don't pursue state of the art anymore, right? So your model will not be able to compete with our models because the size, the training data we have.
But don't you think it's the other way around? But I don't agree with that at all. I think in academia, there's so much ingenuity and creativity, but how can industry catch up?
Yeah, that's we have to stop here because we reserve 30 minutes if you answer other questions.